Okay, perfect. All right, everybody. I'm Dr. Jennifer Geddes McAllister, and it is my honor and privilege to introduce to you Dr. Tamara Doering, an MD PhD from the Washington University Medical School. And Dr. Doering earned her bachelor's degree with honors from Johns Hopkins. And then after completing a medical scientist training program at Johns Hopkins School of Medicine, was a postdoctoral fellow at the University of Cal Berkeley, and then a visiting scientist at the Albert Einstein Medical College, and then an assistant professor at Cornell, and then finally joining the Washington University Medical School. And she is an alumni endowed professor of molecular microbiology. And anybody in the crypto world, it's a nice, small, friendly field. And so I'm sure you've come across Tamara's work. And Dr. Doering is also a fellow of the American Association for the Advancement of Science, a fellow of the American Academy of Microbiology, and a member of the American Society for Clinical Investigation. She served as a regular reviewer for NIH, editorial board members and reviewers for many scientific journals, and on the advisory board member for the Burroughs Wellcome Fund. Among other honors, she's also received a Burroughs Wellcome Fund Award, which is highly prestigious, and this was in biomedical sciences and in molecular pathogenic mycology. And any prestigious award that goes to a mycologist is absolutely thrilling and wonderful celebration. And so I invite you to please welcome Dr. Doering, and thank you so much for accepting the invitation, and we look forward to your talk. Thank you very much.
Okay, well, thank you, everyone. I have to say, I appreciate this opportunity. It's really exciting to have a whole audience that's actually interested in mycology. We don't have to do quite as much education. So what I thought I'd do today is I will tell you a couple of stories, pulling together some published work, and then a new study that we're just writing up. And if anyone can't hear or can't see the slides well, please speak up, and otherwise I will launch. Don't hear anything, so I guess we're good. Okay, so because you're in the program you're in and have colleagues like Jennifer, if I can, yes. You undoubtedly already know that Cryptococcus neoformans kills something like 180,000 people worldwide every year. And that it does this by sort of the following pathway. Just by way of quick review, we know that the disease of cryptococcosis is contracted when a mammal inhales the infectious particle, probably a spore. This sets up a pulmonary infection, and this is a very common event because it's ubiquitous in our environment. In any case, the pulmonary infection resolves, or how it resolves depends on the host, where if the individual is immunocompetent, it's generally asymptomatic, although there's probably latent infection. But if the individual is or becomes immunocompromised, the organism can disseminate throughout the body. And the really devastating outcome is when it gets to the brain and causes a frequently lethal meningitis. So the impact of this is substantial, partly because we've got not great survival rates, even with therapy. And so that gives rise, maybe 1 million cases a year, giving rise to the mortality rates that I just cited. And as you can see on this map, the infection primarily affects areas of sub-Saharan Africa, parts of Asia, really places where you have relatively large populations of immunocompromised individuals, usually due to HIV infection, in the context of limited healthcare resources. And this really sort of global health impact is part of why we study this organism. And of course, another part probably shared with many of you for your favorite fungi is that it offers really interesting biology. And I'll give you some taste of that, I hope, today. So again, just by review, anyone who hasn't thought about crypto too lately, this is a haploid budding yeast. We know it's a facultative intracellular pathogen, so it grows happily in host body fluids or out in the environment as a free cell, or it can also proliferate and do quite well inside of host cells. It's a basidiomycete, and the genome is about 24 megabases, which will perhaps come up later. Now, perhaps the most sort of well-known distinguishing factor feature of this yeast, which was first described back in the 1890s, is its polysaccharide capsule that surrounds the cell wall. And what you're looking at here is an India ink stain. Many of you have probably done stains like this. And what you see is this gray background. These are the moving ink particles. And then between those particles and this structure right here, which is the cell wall, is this sort of halo. And this is the space where the capsule fibers outside of the cell wall are physically displacing the particles of ink and giving rise to this space. So I think you can appreciate that this is a large structure, can be a large structure, and the interactions of this organism with its environment, whether that's out in pigeon droppings or inside of a host, need to take this structure into account. And we can also visualize this sort of up close and personal. We've done some imaging right at the edge of the cell. This is work a while ago with John Heuser. And what you're looking at here is a quick freeze deep etch image. This is the plasma membrane at the left. I hope you can see my pointer. These are actually proteins in that membrane. It's bounded by the cell wall. Of course, this is yeast, and it's got this two-layer structure you can kind of appreciate here. And then emanating from that structure, going out, way past the bounds of this screen and your screens, are these beautiful capsule fibers, and they're arrayed both over the parental cell and also over this very newly forming beautiful bud right here. And so I've been admiring this structure for a long time, and also thinking and trying to understand how it is made, which we will touch on a little more later. But I just thought, sort of by way of overview of the types of problems that my lab addresses, is that it's probably the most long-standing question in my lab, which is how capsule is synthesized. We've also studied the synthesis of other glycoconjugates. So that's been a really long-standing major area of work. We're also interested in interactions between these fungi and host cells. And of course, those interactions often come back to surface structures like the cell wall or capsule or membranes. And we're also interested in how they cross the blood-brain barrier. But what I'll talk about today is sort of two more recent efforts in the lab that have to do more with what's going on inside the cell. And these have to do with the regulation of capsule, and also how we're trying to study genomic variants to understand which ones are important in virulence. So that's right. So that's the plan for today. And to get started talking about these, I think I'll first go back to the capsule, which you can see here stained in blue outside of the green cell wall. And what we know about the capsule is, first of all, it's required for virulence. So cells that don't make capsule or have defects in capsule don't cause disease. We also know that it's unique among fungal pathogens, although there's non-pathogenic relatives that make similar structures. On a sort of in practical terms, on the positive side, this material is used for diagnosis of the disease because besides being displayed on the surface of the cell, it's also shed into the medium. And this is detected by antibody-based tests, and that's how we know and follow the infection. On the negative side, both the material on the cells and shed from them into the host environment has a variety of mechanisms by which it impairs the host immune response. So I've been talking about this polysaccharide capsule. I'll show you briefly what it looks like, even though that's not our main focus for today. And I seem to have lost, oh, there's my pointer. So it's made up primarily of two polysaccharides, and they're cartooned here. One of them is called glucuranoxylomannan. That's because of what it's made of. So it's a mannan because it has this backbone of mannose residues. And then it has side chains of glucuronic acid and xylose. And this is one repeat unit that repeats many of times to a polymer that's millions of molecular weight. And this is about 90% of the capsule. The rest of the capsule is a smaller, maybe hundreds of thousands of molecular weight polymer that's a little more complex. And what you see is this is now a galactan. It has a galactose backbone with slightly longer side chains consisting of more galactose, and then the three sugars I've already mentioned, mannose, xylose, and glucuronic acid. Again, a repeating polymer. Both of these have heterogeneity, especially in terms of the amounts of xylose they bear and acetylation. We won't go into any of that today. And they also both have been implicated in as having a role in infection. So again, we're thinking about capsule regulation, which means we have to think about its synthesis. And I just want to remind everybody that glycan structures are not directly encoded in the genome. And I know everybody knows this, but you may not always think about it particularly. But unlike cellular macromolecules like proteins or RNA or DNA, there's no template for glycan structures, and there's no sort of unified machinery to generate them. So instead, these structures are produced by the sequential action of groups of enzymes called glycosyltransferases, which means you have to think about synthetic processes and their regulation a little bit different. So when we talk about capsule biosynthesis, the steps we might be thinking about, and these are all things my lab has studied over the years, is that first of all, you need to provide the precursors to build this material. And usually those precursors are nucleotide sugars. And they then have to be supplied to the processes that actually assemble these different sugars together to build the polysaccharides. So they often have to be transported into the right subcellular compartment for this kind of assembly. And since we're talking about capsule that ends up on the surface of the cell, we also have to think about steps in export and, and also association with the cell surface. So there's really a lot of steps that we're thinking about. And when I talk about capsule synthesis, I just wanted to point out that I'm really considering all of these events. It's quite a complex process. Now, I keep saying it's regulated. Let me show you evidence of that. Here on the left, we have some Cryptococcus cells in India ink. And you probably noticed that you can't see any of the halo I spent some time pointing out before. However, if we take the exact same strain of cells and now just grow them in a different medium, and I'll say this is a host-like medium, and when I say that, I mean it's tissue culture medium for mammalian cells, 5% CO2, 37 degrees, we now within some hours, see the elaboration of these big capsules. It's the image that I showed you before. So this regulation, of course, can happen at lots of levels, right? So it can happen at the level of transcription or mRNA stability or localization. It can be the level of protein synthesis or post-translational modifications, right? So this regulation could be happening and probably does happen at more than one of those steps. But a few years ago, we decided to sort of look a little more formally at what was involved in inducing capsules like this. And one experiment that we did was to use the protein synthesis inhibitor cyclohexamide to ask if you need protein synthesis for this process. And so what you're seeing here is when we grow cells in the presence of cyclohexamide, and this is 8 and 16-hour blocks, you can see that they're really not making any capsule, even in the same conditions as these untreated cells. If we let them grow for a while and start to make a capsule and then add cyclohexamide, the process stops. And in contrast, actually, if we treat them for a while and then wash them into fresh medium with no drug, you can see there's even a little bit of a rebound. It's kind of like they overmake the capsule. But in any event, this makes it pretty clear that you actually need ongoing protein synthesis throughout this process of capsule expansion. We also did a similar experiment to ask about RNA synthesis. Now we're using 1,10-phenanthroline, so that's RNA synthesis inhibitor. And you really see a similar picture. So you definitely in the presence of the drug, there's no capsule expansion. Although you don't get quite the same rebound. These cells, I think, are sort of more severely perturbed by the addition of this drug than cyclohexamide even. So you don't quite see the recovery. But in any event, I think together, it seems pretty clear that both of these are needed. And this suggested to us that maybe a good place to start looking would be maybe the dominant influences occur at the level of transcription. So if we take a transcription-centric view now of capsule regulation, what does that look like? What it means is we're trying to understand how the sort of stimuli that are sort of coming in from the environment to the cells when they've been perhaps in a change of environment, like going into infect a mammal, these stimuli are transmitted somehow to a network of transcription factors that will be interacting perhaps with each other, as well as with key target genes. And then the product of those genes will essentially collaborate to generate the right size and nature of capsule for whatever situation the cell finds itself in. So for the purposes of the first part of this talk, I'll be focusing on our efforts to sort of understand what's going on in this blue box and giving you a few examples of interesting transcription factors that we have come across. Okay. So we are not, just to make clear, we're not the only people to ever think about these questions of capsule regulation. And we know that in fact, multiple pathways act in this process. And a lot of data has been identified by the labs I've listed and others, mostly by perturbing some signaling pathway of interest. And these are just some of the examples that have been perturbed, and then observing that the capsule is a different size, or there's something different about capsule at the end of that process. So we at least have an idea of the influences that might be occurring on this process. One thing that had not really been addressed before we started working on this some years ago was how these various regulatory pathways interact with each other, which we know must be happening, and is also very important to the outcome of, generating the correct capsule. And so we sort of decided that what we needed to do was really map the transcriptional network. And I'll admit something. What we decided to do was map the transcriptional regulatory network to try to understand these events better. And when I say we, I'm talking about a longtime collaborator, Michael Brent at our institution. Okay, so how do you map a transcriptional network? This is what I learned from the Rent Lab. There's usually a couple of different approaches. So one approach is that of looking at differential expression. And what would happen here is maybe you perturb some transcription factor and then look at the effects that perturbation has on the expression of other genes as evidence of a regulatory relationship. Okay, and this is really classic. You've all heard of stuff like this. This is, kind of who's upstream of who kind of genetics. Another approach is a more correlative one. And here you might take pairs of genes, creatively called A and B here, and look at their expression over a lot of different conditions. And some of these conditions could be the perturbations shown on the left side, for example, but or different environmental conditions. And again, if you see a close correlation between the expression of a pair of genes across many types of conditions, again, this suggests some kind of regulatory relationship, which would still need to be defined. In any case, these are usually approaches that are used separately, but a while ago, a student of ours developed a pathway inference algorithm called NetProfit that would combine these two types of data. And this worked really well on sort of benchmark data sets in Saccharomyces cerevisiae. So we applied it to a lot of expression data that we gathered from Cryptococcus neoformans experiments. And that included a lot of experiments with cells missing transcription factors, so we could take advantage of both arms of the pathway. And what we got out of this was essentially a model of the relationships between the several hundred transcription factors predicted to be in this organism and their target genes. And I'm not going to go into this work anymore now. It's been published a while ago, but I will say it's been really useful in understanding both the interactions between regulators as well as how they mediate their effects on cells. So just to go into this a little more, I'll tell you one more bit of sort of approach that we used is usually when we sort of look at this network mapping idea or our model, what we're kind of asking is, well, what are the target genes of a transcription factor, and looking downward sort of through the sequence of events. And this has been really useful, as I said, in understanding a lot of cryptococcal biology. But another thing we can do is sort of turn this around and say, well, let's look at the gene sets and try to surmise information about the transcription factors. And another graduate student, Zeke Mayer, a while ago, took an approach like this. And what he did was he looked at target gene sets of transcription factors, and he looked at their level of enrichment for genes that had already been implicated through other studies in making capsule or altering capsule. And this allowed him to then rank transcription factors by how implicated he felt they were in the whole process of capsule synthesis. So we looked at the top few dozen genes on his list, sort of going rank order down from the top. And I was really happy to see that quite a number of them actually had already been implicated in capsule synthesis. So that was, very encouraging. And what we then did was made deletion strains corresponding to all of his top predictions that had not been previously studied. And at the time, I was really quite astonished, as well as delighted to see that, that these pretty much all of these mutants had some kind of change in their capsule. And I've lined them up in this gallery by sort of the smallest capsule. Here's wild type in the middle for reference, all the way up through. So there were cells that had aberrantly small capsules, all the way up to cells that had extremely large capsules. And of course, these images were chosen to match the means of, populations of hundreds of cells that were measured. But in any case, this was really quite gratifying, because even though in the years since we did this work, people have developed mutant collections. So the Bond and Madani labs have made collections of cryptococcal knockout strains, for example, so you could go screen and look for capsule. At the time, those weren't available. So it was really this computational modeling that was pointing us to the important biology. And that was really quite exciting. So what I'm going to do next is tell you a couple of short stories about a few of these regulators. But what I want to do first is just mention that we're still also working on sort of the big questions in capsule regulation. So we're refining this network model based on a lot of kinetic data, especially that we didn't have incorporated before. And we're also using sort of different specific environmental perturbations to try to understand, what are the key stimuli that go into changing different pathways of communication to the cell to generate different capsule. And what we'd really like to understand is sort of what are the key regulators and targets and decision points here. So, so that's ongoing work, but I'm going to tell you about a couple factors that we've actually studied a little bit more. And first, I'll just tell you that we took this set of transcription factor mutants and took a look at them in various phenotypes, but I'll just show you one. And this is what happens in a mouse after you infect the mice intranasally with each of these strains. And so these strains are lined up by capsule size. And what you're looking at is the fold change in the lung colony forming units, so the organ burden in the lung, at a week after infection. And here's wild type again in the middle for reference. And what I think you can readily appreciate is that there's mutants, both ones that have small capsules and also ones that have big capsules, that are clearly have altered ability to grow in the context of a mouse. And we use this as a surrogate for virulence. And we were particularly interested in some of these so large capsule strains, because this kind of mutant had never really been picked up before for technical reasons in how people have looked for capsule mutants in the past. So I'm going to tell you a little bit about this mutant, USV101. And as was shown on the previous slide, this has a large capsule compared to wild type. And we can confirm that by making the complemented mutant, and an overexpressor even has a slightly smaller capsule. It's consistent finding. So clearly this transcription factor is influencing capsule. And I've told you that's like a big virulence factor in Cryptococcus, which this group probably already knew. Another virulence factor that's been studied a lot in Cryptococcus is melanin. And so we took a look at that. And what we find is that this mutant also has a defect in melanin. And that's whether you look at the amount that's sort of released from the cell into the medium. In this assay, you can see it's down, comes back, and the control's an overexpressor. This is a strain that doesn't make melanin. And you can also see it very clearly just looking at colonies growing on a plate as well. So given that these cells had problems with two different virulence factors, we thought we better look at them in a more detailed mouse experiment. And so now you're looking at a survival experiment. Mice are infected intranasally the same way, and then we follow them over time. And so we find that if they lose weight to below 80% of their maximum weight, then they're sacrificed. That's considered an endpoint, as well as signs of infection. And what you see here is that in about 3 weeks, all of the mice infected with wild type complement or overexpressing strain have needed to be sacrificed already, where the USV101 infected mice were doing just fine. But we like to take our experiments out a little farther than this. And we then kind of got a surprise, which was these mice did start losing weight and eventually all succumbed to the infection, in contrast to an acapsular strain here, which, the mice are off fine forever, or as long as you're willing to keep checking them. So besides the kinetics of this process, which are obviously very different, another difference that we saw was how these animals were getting sick. So all of these mice in these groups over here, right around the time they need to be sacrificed, may start showing signs of infection that have to do with the meningitis. So maybe they have a little trouble moving around, or they're a little low activity, some motor problems. But in contrast, these mice were running around just fine. But my technician commented to me that he thought they were breathing a little bit hard. So we thought that was interesting. Maybe we better look at organ burdens to try to sort this out. And going back to the wild type here, what you see over time of infection is that the colony forming units in the lung just go up until the mouse needs to be sacrificed, similar for the brain or the spleen, lower levels in these organs. But in contrast, when we looked at the mutant, what we saw is a similar pattern, but everything was really much slower, happening later to detect these counts in the other organs, and never really reaching the same levels as wild type. And so that tells us that it isn't sort of the fungal burden itself that's causing these mice to succumb to infection. And so what we did is we decided again, well, let's look in a little more detail using histology. And for an example, just at the top left is a picture of an uninfected lung. And what you see here is really nice open airways, lots of air spaces, and this beautiful lacy appearance. The wild type mice, a couple days before they would need to be sacrificed, what you see is something quite different now. These are cryptococcus cells you can see staining here in the maroon. And then there's a little bit of inflammation, but the really dramatic observation, I think, is a sort of wholesale destruction of the lung architecture. Clearly, this is a problem. Now, if we look at the mutant, we see something different. So at the same 18-day time point, what we see now is there's actually a lot more inflammation. We see plenty of cryptococcus cells, but there's still areas of the lung that look sort of preserved. And these mice, again, are functioning fine. Toward the end of their lives, however, what you start to see is this massive inflammation, as well as lots and lots of cells, as you can appreciate in this inset. These are all the fungal cells completely filling up the lung. So overall, these animals are essentially dying of pneumonia, of this sort of extreme inflammatory response that eventually is going to make it impossible for these lungs to function in gas exchange. So another thing we were curious about, though, this was sort of focusing on the lung, but if you look at the brain, you can see that the sort of lower burden is actually even greater. And we thought, well, this might be just reflecting the lower lung burden overall, but maybe there's something else going on. And we know that to get into the brain, Cryptococci have to cross the blood-brain barrier. So we decided to take a look at this. And we can model this blood-brain barrier traversal using transwells. And so these are wells where you can put medium in two compartments that are essentially separated by a permeable membrane. We can grow brain endothelial cells on that membrane. And then if we add cryptococcus cells to the top chamber, we can ask, do they get through it? Into the bottom chamber, we can sample and plate for CFUs. And when we did this experiment, what we found was that the USV101 transcription factor is really quite impaired for migration into the brain relative to wild type. And here we again correct with the complement and have maybe even better transit of the overexpressing cells. So kind of putting all of this together, what we feel like is going on is that really absence of this transcription factor is completely transforming pathogenesis of Cryptococcus in this mouse model. So through a combination of slower growth and inefficient crossing of the blood-brain barrier, the onset of the meningitis is really delayed. And meanwhile, the host response is creating the situation of a lethal pneumonia. So that was kind of an interesting story. It definitely was not what we expected when we started studying this particular mutant. And I want to just tell you very more briefly about another one of these mutants called PDR802. And it sort of brings forth another interesting facet of cryptococcal biology. So again, this mutant is a hypercapsular strain. So it has this big capsule. You can, again, complement it back to normal size. And when we looked at lung burden in similar experiments to what I've shown you, what we found is that in contrary to the wild type and complement that keep going up in terms of lung burden, the levels of this mutant stay pretty much the same in the lung. And this is very close to the level of inoculation. We also did a survival experiment, like what I just showed you here, the controls. And what we find is, while a minority of these cells do succumb to the infection much later, many of them are quite fine in the long term. So often, maybe like some of you, when we have a sort of a new mutant and we just start with some basic characteristics, how does it grow on different media? We might do like, serial dilutions in different medium conditions and temperatures. And so the student worked on this, Julia Hofstadt, started doing studies like that. And she did a lot of them, which I won't go through because she never found any defects. And this was really kind of puzzling, especially when we were trying to use sort of conditions that are perhaps like stresses that might be encountered in the host, like oxidative or nitrosative stress. But really, we had sort of no clue what was wrong with these cells. So then she decided that she needed to maybe approach a little more of host-like conditions. And so what she did was to grow these cells, again, tissue culture medium, 37 degrees, 5% CO2, and then sample them over time. And what she found was that while the control cells grew, fine, the PDR802 mutants rapidly lost viability in these conditions. And interestingly, in wild type cells in these conditions, expression of the PDR802 gene, climbs way up. So clearly it is important as a transcription factor, but only in these specific conditions, which are of course of great interest to us. The other thing that Julia noticed when she was sort of looking at these cells for these experiments was that she saw a lot of big cells. And this is something that Cryptococcus cells do. They make really big cells during infections. And this is an image from Kirsten Nielsen's lab, which has studied these along with other labs, and they dubbed them Titan cells for their size. And these cells are really big. They can be, 100 microns across, whereas a normal Cryptococcus cell seen here inside some phagocytic cells is more like, 4 to 7 microns across, something like that. These cells also disseminate quite poorly, perhaps because of their size. And interestingly, they're polyploid, so they can get up to 64x in their DNA content. They furthermore are very good at resisting killing and stress. So some of this may be size. I mean, a phagocyte is going to have a lot of trouble dealing with a cell like this in terms of trying to internalize it. But also they have differences in their cell walls and probably their capsules. They also tend to produce, even though they're so large, produce small progeny. And these can be heterogeneous. They can be, haploid or diploid, or, sometimes a little bit multiploid, polyploid. But the combination of this sort of, these great big structures that are very resistant to damage and produce little progeny has actually led Kirsten's lab to depict them something like this, which I think is rather nice. But we were interested in whether these were really Titan cells in our experiments. So Julia did some studies, both in vitro using conditions that are known to favor Titan cell formation, and also taking cells directly from mice. And first of all, when she studied them, she found that these big cells were indeed polyploid. They fit all the definitions of Titan cells. And they were definitely more abundant, both in vitro and in vivo, in the mutant strains rather than wild type. And she's quantitated this here for mouse infections. So these are the days post-infection. And you can see that the fraction of the population of these large cells is much, much greater in the mutant population. So we've also trying to understand sort of the mechanisms of the phenotypes and what we're seeing with the loss of this transcription factor. We've done some ChIP-seq studies and RNA-seq studies trying to look at direct and indirect targets of this transcription factor. And this was published recently, so I won't discuss it in any detail, but I'll just mention that among other findings, we found that PDR802 is a direct positive regulator of some quorum sensing proteins, which usually act to inhibit the formation of Titan cells. It's also involved in regulating genes whose products are required for responses to stress, for capsule production, and for cell wall integrity. So we're able to explain many of our phenotypes. And the last point I'd make on this particular story is just that it really points out the balance that these pathogens always need to be keeping. So you might think sort of my first principles, wow, these Titan cells are so rugged and they're really a great thing for a pathogen. But in fact, that really may not be true. It might be sort of too much. And you can think of a similar because they can't disseminate. Similarly for capsules, you want to hit the sweet spot where the capsule is small enough that maybe the cells could be, you don't want them too easily destroyed by the host, but on the other hand, they still need to be able to disseminate. So it's just sort of a reminder that when we sort of think about these characteristics and the value they have to the pathogen, it's probably not a question of extremes, but really of the balance, and this balance can shift, especially in the context of a host. So now I'm going to switch gears. I'm going to stop talking about transcription factors, and I want to go on to a short story about something that's kind of new in the lab. And I'm actually going to do this a little differently, and I'm going to start at the end of the story. So what you're looking at here, obviously, you've seen a few of these now, is a mouse survival curve, time after infection. And I'm just going to tell you these two curves are obviously different. And the difference between the two strains that were used for infections here is a single small nucleotide polymorphism, or SNP. So in itself, maybe that wouldn't seem so exciting. But the reason I'm excited about this result is that, our attention was drawn to this SNP by a completely sort of unbiased method, which was just sequence-based, and it was independent of any prior knowledge of this gene or its function. But now, let me back up and explain a little more. So for way of background, we know that distinct strain lineages of Cryptococci, for example, when people have collected clinical isolates, are associated with a lot of different, they have variation in a lot of phenotypes, including clinical outcome, perhaps not surprising. We also know that natural variation in sequence that's found in strains is associated with changes in virulence. And this has been observed both in human populations and in animal models. So this made us wonder whether it would be possible to, without any pre-existing information, identify natural, sort of key natural variants that influence virulence, really just using sequence analysis. And again, this is a project we're doing together with Michael Brent. So why might we want to do this? So there's a few reasons. One is we might identify new potential targets for antifungal therapy, maybe that hadn't been recognized as playing a role in virulence earlier. Another is that if we know the specific changes in genes or regulatory regions, maybe that can help us understand the mechanistic basis of their effects, which is a contribution to basic science. And finally, maybe very speculatively, if we can identify variants that are associated with greater virulence, maybe that would allow us to survey cryptococcal infections for such changes. So I think we have good reasons to look for these variants. Where might we find them? So first of all, we might find a variant that influences virulence in a known gene. And that's actually the story that I'll tell you today. But that's still going to give us information, potentially mechanistic information, rather than just knowing this gene is important if we know specific sequence changes that make a difference in its function or impact on pathogenesis. We might also find variants in novel genes, which would be great. That could direct our attention to them and maybe uncover new biology. And also, unlike approaches where people use the type of gene deletion libraries that I mentioned, or mutation to study virulence or role of genes in virulence, this unbiased approach can allow us to find perhaps essential genes or regulatory sequences, or variants in them that are important, which you wouldn't pick up by those more traditional methods. So we decided to go ahead and tackle this problem. And I should just mention, if you're trying to study natural infection, you run into a couple of challenges. So one, of course, is patient heterogeneity. We know that patients differ in terms of their genotype, in terms of other conditions they might have, and certainly in terms of the healthcare and treatment that they receive. So to avoid this whole issue, we decided that we would look at our clinical strains in the context of mouse infection, and studies, again, from the Nielsen lab have really validated this as an approach. Another challenge comes up with the pathogen side, is that there's sort of limited recombination that has occurred in clinical strains. And so here what we decided to do was take advantage of the fact that you can do genetics in Cryptococcus, so you can cross strains and generate recombinant progeny that might be important to analyze. And what we decided to do was cross clinical strain to laboratory strains, or one laboratory strain, various clinical strains. So first we had to pick a clinical strain to use. And what we did was we went back, we had about 70 clinical strains in the lab, clinical isolates generally given by colleagues like John Perfect. And what we did was first use our sort of organ burden assay. So we're looking at the left here at the log twofold change in lung burden after a 9-day infection. And everything is plotted relative to KN99, which is our wild type lab strain. And what you can see is you have a whole range of sort of virulence. So we're using this lung burden as a surrogate for virulence. So I'll interchange those terms. And the one we decided to use was this strain C8. It had a couple of advantages. First of all, it crosses quite well, which is not true for all the clinical strains. It had a big difference in virulence from KN99. And also when we look at these two strains, they have about 50,000 sites in the genome where they differ in sequence. So single nucleotide or small insertion or deletions. So we went ahead and crossed these two strains, and then we collected the progeny, isolated them, and then we did the same kind of experiment again. So here we took a selection of progeny strains again, put them in mice to look at lung burden. And I want to point out one, I think, important feature here, which is that if you look at where the parent strains are on this plot, now you'll see that the whole population has a much broader range. Excuse me, it's way outside of the parents. And I think, what this is telling us is that each of these parents carries both beneficial and deleterious alleles in terms of ability to colonize a mouse that have fairly large effects. So when you recombine these two genomes, you can get a much broader range, and this will come up later. So once we had this population of recombinants, we turned to an approach called bulk segregant analysis. And what we did is we took pools of recombinant strains, actually larger pools and overlapping pools of recombinant strains, and then we grew them. Then we either sampled them directly, or we grew them in two different contexts, rich YPD medium in the lab, or infected them into mice. We could then recover the organisms and sequence their DNA sort of as a population. And the rationale here is that if there's some region of the genome in which one allele from either parent favors growth in a specific environment, then the recombinants that happen to get that region in meiosis will probably grow better in that environment. And those alleles in turn will be enriched in these final DNA sequences. So what this is really allowing you to do is look at individual genetic loci that differ between the parents and give a growth advantage rather than looking at individual strains. So we can plot these data by looking at the change in allele frequency. And here we're just plotting along a chromosome and arbitrarily calling the negative values the lab strain and the positive values the clinical strain. And if we first look at the data for YPD, so the dots are the mean and the lines are the range of values that we obtained, what you see is that going along this chromosome, this is chromosome 2, but it looked the same for the whole genome, there's really not a lot of action, right? So growing these cells in YPD is not really changing the frequency of any alleles in particular. However, if we now look at the results from growing these pools of cells in a mouse, we see something very different. So I think you can now appreciate that there's regions of the genome where having the clinical allele seems to be a great advantage in terms of survival in the mouse, and others where it's more of a disadvantage. And we were particularly interested in this highlighted region, both because of the sort of the magnitude of the change here, as well as how tight all these numbers were across and between pools. So we were pretty excited about this, but we then wanted to use a really different approach to sort of test this result. And so we turned to an approach that's more of an association analysis. We went back to our progeny strains, and we now did individual mouse experiments. So one recombinant at a time, so we could look at those data. And then we also sequenced these strains. And then to analyze this, what we did was identify SNPs every 3 to 5 kilobases across the genome. And this is sort of cartooning what different recombinants might look like. And then we could go to each of these positions of each of these SNPs and sort of divide the population of recombinants by which parental allele they had gotten, okay? And then what we could do is assign each of these strains to one of these groups according to allele and calculate the student's t-statistic comparing the CFU scores of the two groups. And so at the end of, so there's some other examples of groups. And then at the end of all this, we get a plot like this. And what you can see is that a very similar region is coming out of this analysis. And here they are lined up. And you can see that even though these two approaches are completely different, right? Pool strains and sequencing analysis versus individual strains looking only at colony forming units. So the rationale and the methods, everything is very different. We got the same region on this chromosome out. And we actually then did some additional studies where we forced recombination in this region to try to sort of narrow it down. And we've ended up with a region of interest that's about 55 kilobases. There's 64 SNPs in it and 18 genes. So we felt like these approaches were allowing us to identify natural variants that influence virulence. And then the question is, how can we validate these changes? How can I convince you this is real? And first we had to decide which genes to look at. And what we did was we looked at both the magnitude of the change of genes in this around and in genes in this region, as well as what differences the SNPs would make to their products. And then what we wanted to do was do a validation by doing a gene swapping experiment. And we wanted to swap only the SNP between the two parents. But as most of you know from your work on fungi, usually to do this kind of manipulation, you need to incorporate a marker, some kind of drug resistance marker. And just that, adding that marker can sometimes perturb the behavior of the strains. So what we decided to do was actually make two strains for testing out of each parent, one that had the drug marker and the sequence from the other parent, and another where we just reintroduced the same sequence again, but it's otherwise identical. And I just mentioned this because you'll see all of these strains in mice experiments that I'm going to show you, but these two, these pairs are really the relevant comparison because they're identical except for that SNP or the SNPs in that gene. And I'm going to show you data on this particular gene here, which falls pretty much at the highest point of this change in allele frequency curve. And so that means we would predict from our data that putting this sequence from the clinical strain into KN99 would enhance its virulence. And this is in fact the curve that I showed you at the beginning, now including the KN99 background strain, just for sort of a reference point. And what we see when we do the manipulation to just add the marker, but without any sequence change, it does bump the virulence down a little bit. The survival curve is elongated. But if we now make a strain that's identical to this one in every respect, except for the SNPs in that gene, what we see is that it not only sort of compensates for that problem, but shifts it to even the more virulent side in a very significant way. And you can see that as well if we look at lung burdens of these strains. And so here again, you're seeing these strains differ only in the SNPs in that gene, and they're differing by about an order of magnitude in terms of lung burden. Again, very significant. So I told you that we predicted that you would increase virulence of the lab strain by changing to the clinical SNP, which is what you see here. And we wanted to also take it back in the other direction. So here you see the clinical strain infection over time. You can see the numbers go up in lung burden and eventually all these mice succumb. In contrast, if we've put in the clinical sequence now, although they start to grow briefly, they're then pretty much cleared. This line represents our limit of detection. So we can kind of shift the virulence in either way. And we feel like this really validates our sort of finding in this region. So the last question, at least for now, is what's the mechanism here? So what's going on? How are these genes, how are these SNPs altering the function of these genes? And of course, this is going to depend very much on the locus of interest. And in this case, we were kind of surprised to see the results we found because they were from a gene that encodes a protein called PDE2. This is a high affinity phosphodiesterase that is known from studies in Saccharomyces cerevisiae and Candida albicans to regulate basal cyclic AMP levels with sort of implications for stress response and other characteristics of the cells. And we know So the reason we were surprised, so that sounds important to the cell, but the reason we were surprised is that work from a while ago from Joe Hyten's lab had published that deleting this gene in Cryptococcus made no difference. So that was a little puzzling why we would get the virulence results we do. So we looked back at the sequence, and in fact, there are 3 SNPs contained within this coding, this sort of gene sequence. One is a short insertion, and then a couple other ones that may or may not change amino acid residues. So we decided to try to separate these out and figure out which was actually important so we could study it further. And one of the first strains we made was one where we had the original insertion from the clinical strain, now in the lab strain background, but the rest of the gene was identical. So the only difference between this strain and our control is this insertion of 4 base pairs. And of course, we've checked all that by genome sequencing. And what we found when we then went back to our lung burden study, now it's a log-based 10-fold change, sorry to swap axes on you. What we found is that in fact, this strain with only the 4 nucleotide insertion looks just like the strain where we've changed all 3 positions, showing that that's the insertion that matters. And again, significantly more higher burden than you see in the wild type strain that that doesn't have that insertion. So then we had to think about this, like we started thinking, why does a short insertion in an intron make a difference? And we're trying to suss this out. And we decided to do some RNA-seq to try to get some insight here. And what we found was that, in fact, this is not an intron. So this region is robustly expressed in both the clinical strain and the parent strain. And moreover, in the lab strain, in the lab strain that doesn't have these 4 nucleotides, this would then be in frame to form 2 stop codons. And in contrast to the clinical strain that would happily make the entire protein with an active site that's down near the C-terminus. So what we actually think is going on in our model is that there's no functional PDE2 in the laboratory strain at all. We think this explains the difference in, sorry, got a timer coming up. We think this explains the difference in virulence that we see. And we think it's also why there was no change in virulence when this deletion was made before. So just to sum up, I think, it's less about PDE2, in fact. And the message I more want to say is that if you take an unbiased view, and step back, I think you can really reveal some, new biology, some of which you couldn't find by traditional approaches. And beyond PDE2, we have a lot of interesting candidates to explore. In the course of this work, we've also generated resources in the form of hundreds of progeny strains from lots of clinical crosses, many of which are already sequenced. So these are a potential tool for studies. Anytime the parents differ in a trait you're interested in, you could potentially take this approach, and I'd be happy to talk to people about that further. But I will stop here. Sorry, I ran a hair long. And again, I acknowledge that all of this work was done in collaboration with Michael Brent in our Department of Computer Science and Center for Genome Sciences. The transcription factor work that I mentioned early on was done mostly by these prior lab members, as well as a current staff scientist. The later project about PDE2 was really done by this postdoc, Daniel Agustino, with the help of a staff scientist and a couple of undergraduate students in the lab. And I also need to, of course, acknowledge the rest of the members of the lab who are working on other cool projects, as well as our funding from the National Institutes of Health. And I would be very delighted to take any questions. Thank you.